our last activity of the day is a panel where we have people with expertise in quite wide-ranging areas representing the topics that we discussed today. And really this is a chance for us to brainstorm and think and question anything to do with what we can do in the realm of research to do with water around One Health. Everything from employing the power of fish models to thinking about environment and disease to even wastewater. Hopefully you guys have some ideas, some discussion question things. We'll have mics circulating around, maybe looking for connections between our different talks. And I'll have to pick on people if we don't have volunteers. Mark, do you have a thought?
It's not a well-formulated question yet, but it's something I've been thinking about throughout the day. How do we incorporate infectious diseases with fish models? And then maybe, as the last speaker, with the climate change happening, we know that there's human pathogens that are making their way to the northern, into the Arctic circles. So I'm assuming human health is changing, but is animal health also changing based off of the pathogen transport? So two sets of questions, one for the fish people, and maybe Chuck, if you know any of the kind of stuff related to how pathogens are changing throughout the environment
as well. I don't know. I just say basically we're screwed. That the pathogens are going to come, and vectors of pathogens like mosquitoes are going to come. They're going to go north, and without dealing with the climate change, that's just going to happen. And in our case, we need to go for that last paragraph that I presented. We'll need to go back there and see if this is increasing, because we don't know if this was a one-time thing. We saw it, and so we're wondering, is it going to continue? We need to go back to Andvert Bay and collect more fish to see if it's there. Is it going other places? Last week there was a paper showing that they found some animals like this. Some Australian Antarctic researchers found some animals, some fish that look like this, and they published a paper all the way across on the other side of Antarctica. So I'm afraid that this might be a general thing, and then to generalize as you are doing, I think it's pretty scary.
Yeah, you know, you look at the animal exposure, but the pathogens take advantage of this too. Naegleria fowleri, the brain-eating amoeba, is a classic example of that. It used to only be the southern states, and now it's in Minnesota, it's in Iowa. As the waters warm up, these pathogens are causing infections further north. Also, as I pointed out, if you hold viruses at warmer temperatures in river water, they become more resistant to disinfectants, it's been found out. The other problem is the natural microflora. Aeromonas hydrophila infections are related to water temperature. So as water temperatures increase, we're likely to see more Aeromonas hydrophila infections from swimming in surface waters and that. So I think surface waters are going to become more of a problem in northern latitudes because these certain pathogens are going to take advantage of them.
I don't think I have anything to add. Yeah.
I think I could just add one thing which relates to your question, Mark, and that is we're seeing large-scale wildlife die-offs in certain areas, like in the Bering Sea, we've had seal die-offs, also in the North Atlantic, seal die-offs, and also we have entire taxonomic groups at risk, like frogs with chytrid fungus infections, bats with white-faced nose syndrome. And of course, those are novel organisms that are infecting these animals, but also it relates to immune function, whether these animals can mount an effective immune response, which kind of goes back to your mixture stuff and kind of work we're doing. If animals are exposed to high levels of contaminants that compromise their immune function, then you can expect these kinds of die-offs. And it's very difficult with these wildlife die-offs because by the time you do the autopsy, they have so many secondary infections and other issues that it's hard to know the causative agent of the original disease. So I think this is an area that you're asking about that we need a lot of work in this area. And it does relate to global change, changing temperatures, changing pollution regimes, all of these kinds of things. So great question. Kelly, please.
I'm wondering if I could get some feedback on maybe experimental design or just the likelihood of using a fish model for studying something that has really differential impact in the human population. Say, for example, mold exposure, so that increases with climate change and flooding, more mold potentially. But not everybody is sensitized to mold toxins. And then how would you potentially look at that relationship that's theorized between Lyme disease and then increased sensitivity to mold toxins and neurological or kind of cognitive impacts of mold toxins? Any idea about fish models for that, or how you would study those types of interactions?
So I don't know of studies that have been done on fish with molds as pathogens. Maybe some with some yeasts, but it's easy to put things in the water and to expose the fish, and there are reporters for the immune system function, and so we have fish where their neutrophils are red with a red fluorescent protein marked with the red fluorescent protein, transgenic fish. And so it's very easy to count the number of neutrophils in those fish. There's a few hundred, a couple hundred neutrophils in a single fish, and a machine can count them. And so it'd be pretty easy to do a lot of experiments where you would treat fish with different molds, or treat them with a mold and then some other anti-mold compound or something like that. I could see quite a few experiments doing that, and then counting the neutrophils as a marker for the response of the fish's immune system to the, I guess it's not a pollutant, to the pathogen. And there are also reporters for things that turn on other kinds of stress responses, where the fish turns a color, or certain cells turn a color when different stressors are given. So it would depend on exactly what you wanted to look at. But they're very good reporters.
And Tara has a comment.
Yeah. Just from the clinical perspective, I do see patients who report mold exposure, toxicity symptoms. They've been told by other doctors or other practitioners that they have a disease now because of their mold exposure. And I will tell you that this is something that did not exist when I was in medical school. We learned about mold sensitization and we learned about infection. But this idea that there's some sort of more permanent change because of an exposure to something in the environment that affects your body on an ongoing basis it's been very, very challenging, I think, to understand from a human perspective. I think there are people who are doing research in that area to try to understand what's happening, but I think the research is maybe not very well funded, maybe not very rigorous, maybe just very difficult to dig into and at very early stages. I think it would be wonderful if more rigorous research was being done, because I know there are a lot of patients out there who have symptoms that are unclear and that we can't help for, right? And as a physician, I think that's one of the most frustrating things, is when a patient shows up with a variety of symptoms and I say, I have no idea what that's from and I don't know how I can help you. And so So I would absolutely, support that. I think it would be interesting to look at, I don't know, to maybe more broadly assess human samples in like a multi-omics experiment, just to get a sense of what's changing in these patients or what's different in these patients from people who report health. Because I don't know if it's the immune system. A lot of the symptoms that are reported by these patients are neurologic, right? Or we're not seeing neutrophils and eosinophils and, IgE levels and things like that are markers of immune function different in those individuals than other people. So either the tools that we have aren't good enough or we're not looking in the right direction. So I think it would be fun to do proteomics and metabolomics and single cell gene expression studies and try to put all this together to understand the biology for those patients. And maybe that would be a good time to dig into a zebrafish model and put in the gene sequence that's related to that outcome or that it overexpresses that protein that seems to be abnormal and maybe see how they respond.
And I think the chronic sequelae associated with COVID really kind of made us all more aware of how infectious diseases can lead to really diverse range of symptoms that can be neurological or imbalance in the gut biome. There's so many things that COVID showed us were part of chronic sequelae, so we need more investigation around that. We've known it for other pathogens for a long time, but not a lot of research on chronic sequelae.
But I know there are NIH-funded studies and giant cohorts that have lots of different kinds of scientists and lots of different kinds of doctors studying it to try to understand it better. So maybe that'll be the first step towards unlocking some of the directions to look for these other chronic diseases.
One side of this that I really like to see personally is I would love to see a lot of research into the way that the exposome is influencing susceptibility to, for example, long COVID, right? So we're talking about how some people are affected very severely by COVID and others, it seems, not at all, and what's going on there. I think there's probably a huge role in chronic low-dose exposures. We don't know anything about that. And not to say that an exposure to a certain chemical is causing you to be sick later, but that you would have enough going on through inflammatory cascades or neurological cascades of some sort that you would get a heightened susceptibility to not compensate when you become sick, to not mount the response needed, or to mount an abnormal response. And that seems very, very complex to me and kind of overwhelming. I don't know anything about it, but just as a comment to what you're saying, that would be amazing.
And I think that really gets to the idea of how the environment influences our health, which is what our study is looking at just a tiny little slice of in relation to the asthma outcomes. But the change in the prevalence of some of these chronic diseases, not just in relation to infectious diseases, but other chronic diseases that are complex and with unclear inflammatory bowel syndrome, cardiovascular disease. A lot of them are getting autism spectrum disorder, have been increasingly linked to microbiome changes and sort of differences in the way that our bodies are functioning in relation to the environment. So I think you're absolutely right. I think it's really important to start studying that in a more rigorous way and methodologic way. We have a study called the Tucson Children's Respiratory Study, which was started in 1980. And here in Tucson, it was healthy babies who were recruited, at birth, and we're still following like half of them today. And they're in their 40s. And we have samples dating back to cord blood. And so, this study was designed to assess respiratory health outcomes, so asthma, COPD, those sorts of things. But it's like this amazing repository of samples where we could look back 40 years and say, what's the prevalence of microplastics in somebody's blood? How much of the different chemicals that have been talked about today that might be present in the drinking water, how much of that is present in the serum of these individuals, and when does it start to increase, and does that increase of exposure or increase in detection in the samples relate to the time course where we're seeing these diseases develop? Our cohort's not really big enough to look at all the different diseases, but at least to sort of get a human time course of when these became, at least detectably present in the body.
You have a question?
Yeah, I think I could speak loud enough. You mentioned the utility of fish and other models in spherocytosis, and I'm wondering if those models can be applied or have been applied to other really prevalent hemoglobinopathies such as thalassemia. thalassemia and sickle cell disease?
So none of the yesterday, well, today actually, we had accepted a paper that has the genome sequence of 24 different Antarctic fish, and I haven't gone through all of them yet, but Toma, my postdoc Toma, is going through all of the genes that are involved with red cell pathologies and is looking at all of those genomes now. And the spherocytosis mutations are the first ones that he's seen, and he's seen a couple of them in different species. There are several different genes in humans cause spherocytosis, and he's found a different mutation in 2 different dragonfish species. And for thalassemias, they don't have a thalassemia. They have beta hemoglobin and alpha hemoglobin like we do, but they don't have a specific thalassemia kind of mutation. They don't have a sickle cell mutation. They have the cells where the cell shape is changed, but none of them look like sickle cells, and it's not a matter of the hemoglobin molecules piling up together to make long rods that cause the cells to change shape like sickle cell disease. But I think that there will be other, when Toma finishes looking at those other sequences, that he will find other mutations that are similar. And he may find mutations in other genes that may help explain other fish genes that may have orthologs in human, where it's not yet known whether this particular gene causes a hemoglobinopathy in human, and it may suggest that it does.
So a follow-up with that would be, do you foresee actually targeted biologics that could essentially alleviate these at the basic level?
Yeah, so for the ice fish that has this profound anemia with no mature red blood cells at all, it gets along basically for 2 or 3 reasons. One is because there's more oxygen in the water because it's so cold. 2, because of adaptations that they've made with respect to the carrying of oxygen just in the fluid in the blood, by making the very large heart. The heart is about 3 times larger in the ice fish for body size than in the red-blooded fish, and the third is the bore of the capillaries, which is they don't have capillaries. They have very large bore blood vessels, and none of those really seem to be good therapeutic options for a human with an anemia, but there may be other things that might turn up.
Yes.
As an outsider, if funding was not an issue, what in the next, say, 10, 15 years would be the most important scientific discovery, breakthrough, whatever, that could happen with water that could help everybody?
We need to really completely think how we treat drinking water. Our recent research and recent evidence indicates that current technology is not removing these viruses that can repair themselves in that.
Right.
And I'm really concerned. We've been producing reclaimed water and drinking water for decades, and these viruses obviously are always present and potentially causing endemic infections all the time. I think how we treat drinking water needs to start with understanding molecular mechanisms by which viruses are resistant and how to get rid of them. That's not only from human health, but animal. The requirements for treating water for animal consumption is less because we assume they didn't get infected with human viruses when those regulations were developed. That's not true. We now know that's not true. So I think there has to be innovation in engineering technology to produce safe drinking water. The other thing that's going to happen, and I didn't touch on that, is one virus group infects another. The dependent virus, we're now seeing if you're chronically infected with a virus and you get hit with another virus infection, you're more severe. And I think that's, the data keeps indicating that. We're finding more viruses or organisms are related to human diseases. We thought like multiple sclerosis and other diseases seem to have a microbial link. And I think a lot of these chronic diseases have microbial links to them, and not really, and that's what's going to happen in the future. They're going to link those together.
I was going to say end poverty and world peace, but I don't know if money's no issue, right? So go ahead.
Well, I was going to ask, I have this ideal, which I'm plagiarizing because it's someone else's idea, but of a class-based system of chemical regulation where we say, we know PFOA is bad, so we're going to ban PFAS in all products, we're going to remove it from the water, and we're going to say guilty until proven innocent for chemicals. How big of a burden is that going to be economically for wastewater treatment if we made that happen in the regulatory world through policies? Would it be even economically feasible? Is it possible?
The microbial chemical contaminant, the chemical contaminants are easier to deal with than microbial, actually. They're much simpler to remove because they're always the same all the time. Can we remove them all to the levels where we feel there's a risk? Maybe not, but then again, it takes 50 to 70 years to kill you. So I think looking at the stamp, or a microorganism can kill you in 24 hours. That's the thing that World Health Organization always puts forward all the time. That's not to say that removing the chemicals, I don't think we can always remove them to the levels we would really like to. It's really a challenge in that standpoint from that point. But we can do, based on risk analysis, what we can do. I don't know if most chemicals for drinking water are designed to reduce your risk of mortality to 1 in 10 million to 1 in a million lifetime. Type of thing. So our risk exposure is much less for the chemicals than it is for the microbes, to be honest. We tolerate a much higher risk from the microbes than we do the chemicals right now, to be honest.
Can I ask a question? This is a little bit off the wall, but I know in terms of like products and exposures, cosmetics, soaps, that sort of thing, anything that touches your skin is much tighter regulated in Europe and in other places than it is here in terms of having to sort of prove safety or consider guilty until proven innocent, right? So I kind of wonder if the same is true for the way they deal with water, reclaiming water, treating water. Do you know if there are different standards practices in Europe or if it's similar to U.S.?
It depends on the country, but European standards for chemicals tend to be tighter than they are in the U.S., to be honest, than they do. They actually tolerate the microbial risk greater than they do. To give you an example, in the U.S., our waters for sewage discharges have to be fishable and swimmable, right? That's not Europe. The river sewage discharges are often not disinfected because they have no regulations for fishable and swimmable. So next time you think of jumping into a river in Europe, think about it twice.
But at the same time, you can maybe look at the health or the chemical exposures and some of these other markers in different populations and see if there is any relationship with those outcomes that we're worried about, right? At least for the chemicals, not for the microbes. Yeah.
Okay, other questions for our panel or thoughts?
I just want to say that in Europe, there are a lot of countries in Europe, right? Not all of them have the same regulations. Not all of them have the same money, because at the end, when we are talking about sewage treatment plants and clean water, it also, it has to do with the money, because cleaning the water is very expensive. So, yeah.
Just to comment on that, I'll pass it on. To go from sewage to drinking water costs about $2,000 an acre foot. we pay about $260 an acre foot for the water we get from CAP. But people in Yuma pay $9 to $50 an acre foot to get rid of it. So going from producing wastewater to drinking water is expensive.
Yeah, but having sick people is expensive too.
Yeah.
Great. I'd like to thank our panel. Appreciate it. And we have closing remarks from Dean Hakim now, and then after that, for those of you who would like a glass of wine and a snack and so on, we're going to be outside for some socializing for a reception.
Hi everyone, let me start with a land acknowledgement statement that we respectfully acknowledge that the University of Arizona is on the land and territories of indigenous people. Today, Arizona is home to 22 federally recognized tribes, with Tucson being the home of the O'odham and the Yaqui. Committed to diversity and inclusion, the university strives to build sustainable relationships with sovereign Native nation and indigenous communities through education offering, partnership, and community service. So, greetings everyone. We have seen an impressive series of presentations today. Thank you all for being here at our third One Health Symposium. And I am Iman Hakim, the Dean of the Mellon Enid Zuckerman College of Public Health. So we were honored to have Dr. John Postlethwait from the Institute of Neuroscience at the University of Oregon with us today to give the keynote address about using fish models as in One Health research. I learned a lot, that my first introduction to fish research. We were also honored to have Dr. Sharon Megdal from the Water Resources Research Center here at the University of Arizona with us to talk about the concept of One Water. So thank you. So all of us here today and so many others across campus give our heartfelt thanks to Dr. Frank von Heppel for all of the outstanding work he has done to launch the One Health initiative here at the University of Arizona. He has brought together researchers from across the university and around the state to advance our understanding and expertise in the One Health arena. By working together on complex One Health challenges, we will lead to find solutions in Arizona, in our nation, and around the world. So thank you, Frank.
Thank you.
You did it. Also, I would like to take this opportunity to thank all the presenter and panelists. I know I am between you and the reception, so I will not list all of the contributors, but they are, as you have seen, from across the health science colleges. They are from across department and colleges within the University of Arizona, and our own here for public health contribution, our outstanding faculty in One Health as well. So water is life. As I think about it, this is really true. It has been recognized by indigenous population worldwide for ages, from here in the US, North America, all the way to Egypt, my home country. I remember growing up, and my grandmother always to emphasize us drinking water better than any beverage, Coke, or anything else. She will always say that water is life. Without water, there is no life. So what a theme you have picked for today. So the theme of water and One Health focus our understanding of complex health challenges that have serious consequences in people's lives. From the spread of waterborne diseases to the impact of climate change on water availability, it is clear that water is an essential factor that must be considered when addressing the interconnected health challenges we face. And these symposiums remind us how by working together, we will make a difference using the One Health approach. So One Health research is intrinsically collaborative, as you can see across the room, and interdisciplinary. And I hope that the presentation we saw today and the insight from our panelists will inspire us to build connections. connection with our colleagues across the university, connections with other governmental agencies, and connections with our community's organization and programs. So we can work with each other, bring together our knowledge, and solve the complex and interconnected health problem we face today. So again, I would like to give special thanks to Dr. Frank and to Griselda, she was somewhere here,